Well, good afternoon. I want to thank the organizers of the Sachs Neuroscience Conference for inviting Inhibikase to present some of its early clinical and mechanistic insights into Parkinson's disease. We are a publicly listed company in the NASDAQ with the symbol IKT. And what I'll be discussing today are therapies in clinical development to reverse the progression of Parkinson's disease in human patients, something that we recognize is a bold statement to make, but we'll try to illustrate why we think such therapeutic outcomes may be possible. I just need to make a disclosure on the next slide related to the fact that I'm making forward-looking statements. We have a number of public filings, and I encourage anyone interested to learning more about the company to review those filings in their entirety. As an overview, Parkinson's disease is a very large market. We expect the Parkinson's disease market to double over the next few years. It is already a $6 billion plus sales market, and the country with the largest prevalence of Parkinson's disease in the world happens to be the United States. Inhibikase itself has an emphasis on neurodegeneration, but is a multi-therapeutic pipeline company. Our lead programs are focused on developing drugs that target the core participants in a common pathway related to Parkinson's disease itself. And our lead asset, 14809, is being currently clinically evaluated and has been shown in animal models that reproduce human disease to both therapeutically halt disease progression and reverse functional loss substantially inside and outside of the brain. We have 2 INDs that were filed a number of years ago, began clinical studies earlier this year, and have extensive patent protection. One of the things that's unusual about Inhibikase is that we have funded the company prior to our IPO of last year, mostly by looking for our peers to evaluate what the companies are doing and offer its scientific support and critical review. And we've done that through federal contracts and grants with more than 20 million garnered to date. You can see here our multi-therapeutic pipeline. 14809 is a small molecule Abelson tyrosine kinase, or c-Abl inhibitor. It's being applied to Parkinson's disease in the brain, as well as Parkinson's disease in the GI tract, and that is a differentiating feature for Inhibikase. We believe that the GI tract plays a critical role in both understanding disease in the brain. There is evidence that I'll discuss that actually suggests that Parkinson's disease may begin in the GI tract, and more importantly, since the earliest manifestations of disease on the GI, we believe the GI can inform substantially on understanding target engagement, biomarkers, and disease modification. We're also developing an oncology product, which is a method that we're using to develop a prodrug technology, a platform technology for kinase inhibitor delivery, and we'll also have a number of research phase programs that I'll not discuss today. Parkinson's disease, as many of you know, is a very large market disease, about 700,000 to 1 million patients worldwide in the United States, about 15 million worldwide, with an average age of onset of 60 years of age. It is a slowly progressing disease, typically over 1/3 of a patient's lifespan, with 60,000 new cases and 38,000 deaths a year. It tends to be a disease of men over women 2 to 1, not related to a Y chromosome. And one of the challenges in development is the fact that the comorbidities, the diseases that patients already have due to their age and or overall health, are complications that we have to address, particularly cardiovascular disease, where many small molecule drugs could have important drug-drug interaction concerns that we have to address in developing the overall strategy. As many of you know, Parkinson's disease is also going to progress not just as a movement disorder, but as a disorder of cognitive function. Many of these patients go on to full-blown dementias, and about 60% of Parkinson's patients actually progress to one or another form of Alzheimer's disease. And so we think there's a mechanistic linkage between them that we'll touch on later in the discussion. As in terms of causation, for a long time it's been known that a small non-essential protein known as alpha-synuclein is intimately linked to the disease itself. It looks like a cold coil, as illustrated here, has a long unstructured tail that I'm not representing. But in the disease state, this cold coil is reconfigured or misfolded into a stack of planar beta sheet or discs. The stack of disc-like synuclein sheets we refer to as a plaque. And most of the efforts to date that have been used in the clinic to try to address Parkinson's disease have been focused on removing the plaque. And it was reasonable to think that that might be beneficial because the plaque only shows up in the disease state. But I will try to convince you today that in fact, the plaque, despite its being abnormal, despite it being a misfolded protein, in fact, plays almost no role in causation of disease. It's required for disease initiation. But in our view, what the plaque is doing is activating the Abelson tyrosine kinase c-Abl, which I'll explain how and why. And it is Abl activation that's actually driving the disease process within the affected neurons themselves. And so in our view, this is a significant shift in understanding causation in the disease and therefore how we can possibly treat it. So if we were to now characterize at a high level what the mechanism of disease initiation and progression looks like, there are a number of stressors, as we'll call them. They can be environmental of nature because most forms of Parkinson's, more than 90%, involve failures in metabolic respiration in the mitochondrion, and therefore oxidative or nitrosative stresses that affect the mitochondrial function are a primary driver of early disease manifestation. There are a number of point mutants in known proteins, synuclein itself, and proteins like LRRK2 or GBA1 and others. And a number of those proteins that have specific genetic mutations cause rare early onset forms of disease. We think that they contribute to the spread of disease, but not to the initiation of disease itself. You can also overproduce synuclein in some genetic defect cases, which also leads to the formation of these synuclein aggregates. So while we don't understand what the process is that leads to aggregate formation outside of the affected neurons, these plaques, as we're referring them to, represent misfolded alpha-synuclein. They're definitely abnormal, and in our view, they are completely and totally benign. And this is the area where immunotherapies that have recently been shown to be ineffective in Parkinson's disease were really targeting. So in our view, what happens in the disease process is not the formation of the synuclein itself, aggregates themselves, but their uptake by the affected neurons. And when neurons in the substantium nigra and the striatum take up things like these synuclein aggregates, they trigger the a surveillance or sentinel mechanism, and that sentinel is the Abelson tyrosine kinase or c-Abl. And what c-Abl does in any mature cell type is to sense the presence of abnormality and drive the destruction of the cell containing that abnormality so that other neighboring cells are not affected. So you could view the area of the brain where synuclein aggregates are taken up by these affected neurons as a sort of community, and members of the community are willing to sacrifice themselves for the betterment of the whole. And so what we believe happens inside the affected neurons is that when Abl senses the presence of these synuclein aggregates or plaques, it chemically modifies the plaque, and we think this is really what's creating the toxic form of synuclein aggregates inside the affected neurons. But there's a second step in the process that we believe is even more important. That second step in the process involves c-Abl inactivation of a protein known as Parkin. Parkin is a ubiquitin E3 ligase. It plays a critical role in mitochondrial integrity and quality, and it also plays an important role in surveillance pathways whose purpose, or excuse me, survival pathways whose purpose is to degrade abnormal proteins or objects inside a neuron containing them to protect the neuron from any deleterious effects of that abnormality. When Abl engages Parkin, it activates its enzymatic activities, and as a result, you don't have a survival pathway, you have the formation of toxic synuclein, and that neuron is going to die slowly over time. So what's the basis of our belief that much of the action of Parkinson's disease occurs inside the affected neurons? Well, what we've done with our collaborators is to deploy specific animal models that reproduce the rate of progression of human disease relative to lifespan. In humans, it's a 25-year disease span to death. At the midpoint of that 25-year period, you have 50 to 80% loss of the nigral neurons, nigral dopaminergic neurons in humans. And therefore, when we translated that disease to a practical species in the laboratory setting, which in this case was the mice lived for 3 years. Doing an experiment over 1/3 of a mouse lifespan or for 1 year is extremely challenging in hundreds of mice at a time, but it can be done. And when you do that, you get very satisfying information. So here's what the nigral neurons look like in an ordinary mouse brain. In this case, we've introduced an expression vector for an inherited form of Parkinson's disease driven by the clinical mutant synuclein A53T. At the 6-month time point, you can see this is a substantial loss of neural density. When you quantify those neurons by either Nissl or tyrosine hydroxylase or TH staining, you see a 50% loss over a 6-month period of time. So at the midpoint of the mouse disease, we're seeing the hallmarks of neurodegenerative defects that are comparable to what's seen in humans. And so this was one of many measures that were made to validate that these humanized mouse models actually represent the human disease in many of its salient features. So what is then these gray bars? Well, in mice, we can do something that we can't do in larger organisms, and that is we can do conditional knockouts in the brain, in this case, a conditional knockout for the Abelson tyrosine kinase itself. And as you can see, despite the fact that we introduced the plaque, we put the plaque in the right species, right part of the brain, when Abel is genetically deleted in the adult mouse at 6 months, you see no evidence of neurodegeneration whatsoever. So imagine what's being said. You have a plaque, it is only associated with the presence of disease, and yet there is no disease if Abel isn't turned on in the affected neurons. To us, this was a watershed event, first discovered in 2016. And I'll show you as we move through the presentation what the consequences are of Abel inhibition from a pharmaceutical intervention that tries to reverse the effects of these neurodegenerative events. So do these events occur in human beings? We believe they do. If you take post-mortem patient brain from Parkinson or PD patients, stain the synuclein Lewy bodies, run them out on Western blots, you can see that in the Parkinson's patients using an antibody specific for an Abel-modified synuclein, that's a phosphorylated synuclein at tyrosine 39. You only see that in the patient brain. You don't see that in the age-matched controls. So while this is not a complete picture of just how the biochemical processes that are now published about Abel activation in Parkinson's, this is a compelling picture to say that Abel activation is occurring in the disease context in human patients. So if we now go back to our view of the disease process, our intervention, which is an Abel tyrosine kinase inhibitor, we call IKT-14809. What I'm just giving you as a summary is that we can both inhibit Abel itself and block the formation of further formation of synuclein aggregates with tyrosine phosphorylation. And more importantly, as I'll show you specifically, we can clear toxic synuclein, which we define as the Abel modified form. And we believe that clearance is occurring through reactivation of Parkin, and that leads to functional recovery. So what is 14809? It's a small molecule Abelson tyrosine kinase inhibitor. We have what we call a RAMP or re-engineering approach with metabolism preserved medicinal chemistry program that's described on the Inhibikase.com website. In this case, we use as a template for design imatinib, or the well-known anti-cancer agent that is commercially sold in a branded form as Gleevec. Imatinib cannot bind the wild-type enzyme in a therapeutic context and does not treat in the brain, but it had other pharmaceutical properties that we thought were very salient for Parkinson patients. We designed 14809 using imatinib as a template. It's about 18 times more potent against the wild-type enzyme relative to imatinib itself. In rat and monkey pivotal toxicology studies, we were cleared for human equivalent doses up to 600 milligrams, just as a comparison. Imatinib standard of care dose for therapy is 400 milligrams. So this suggests that 14809 may be even safer than imatinib itself is in older or elderly patients with chronic disease. We saw no specific or organ-specific toxicities, no cytotoxicity, and unlike imatinib, we get very robust brain penetration with sustained concentrations at above 1 micromolar. That's more than 10 times the amount we need to saturate the Abel enzyme in patient brain. There's no neurotoxicity. We see no acute toxicity on long-term dosing, even at high doses. We get nearly complete neuroprotection of alpha-synuclein progressive disease in a prophylactic context. And in a therapeutic context, we will review that shortly. We've done commercial scale manufacturing and have long-term patent protection. So we're in the midst of our clinical trials in phase 1. And those trials, which were initially planned to cover in a roughly 13-month period, have recently been accelerated because of the outcomes of early clinical dosing that we made reference to in a recent filing with the SEC called the 10K filing. We are in the midst of single and multiple ascending dose phases that are interleaved in the study about month 3 out of a total period of time of month 7. And at the same time, we're performing chronic toxicology studies, and we're now at month 4 in that regard. And so what will happen probably around the midsummer timeframe is to extend the study into Parkinson's patients as a phase 1b extension study that will require some further communications with the FDA that is ongoing. But we believe that these studies have now accelerated quickly. And as we announced in our recent 10K filing, we've already demonstrated that low oral doses in elderly or healthy subjects can reach the same exposures of the drug substance systemically that we've seen in efficacy models that I'm about to describe. So what we've learned in the clinic in just a few months is that the drug looks to be relatively safe on early dosing trials. It looks to be very well absorbed. It looks to be very well distributed. And we've already reached exposure levels that are consistent with what we saw in animal models for efficacy. But we do not know anything about efficacy in Parkinson's disease just yet. So how do we come to such a conclusion about the importance of the Abelson tyrosine kinase? Well, it came through a series of studies inside and outside of the brain. So we'll look at the brain studies first. In this case, what we do is we introduce the synuclein defect. I'll show you data related to the A53T inherited form of disease, but we've also done it with a model of idiopathic or random disease. So in the inherited case, we introduce a defect in one hemisphere. When you do, those mice are very efficient at traversing circular mazes because one side of their body is no longer coordinated while the other is hypercoordinated. And so if you then evaluate them in what's called a amphetamine rotation test or evaluate how well they traverse a circular maze, what you find is that those mice are very efficient at doing so. So if you now quantify what happens in the disease state, normal mice will do about 25 circles in 10 minutes. Diseased mice at 6 months duration, so 6 months into the disease course, now have 90 circles in 10 minutes, again, because we've only introduced the defect in one hemisphere, so we can use the other hemisphere as an internal control for all our biochemical and neuroanatomy studies. And so you get a very robust outcome. But if you begin dosing near the end of month 2 and read out that at the end of month 6, you can see that those mice now go back to near normalcy. They now do 30 rotations as compared to 25 in the controls. And that's true at different doses and even true when we add other contributory factors. And it's to very high significant digits. So what this experiment tells us right away is that both cognitively and in terms of motor function, therapeutic administration of an Abel kinase inhibitor like 14809 is very efficient at restoring lost function in these animals. If you look at the neural anatomy, the same is also true. Rather than losing half of the neural density in the affected hemisphere, we now only lose about 20%. We are not implying that what's occurring here is the regeneration of neurons that were lost. Rather, what we've do is we've halted the disease progression by beginning therapeutic dosing near the end of month 2. And those neurons that have not fully degraded are the ones that we're preserving. With clearance of toxicity, we're restoring function to neurons that have not fully degraded. And we're investigating further the nature of the neurodegenerative events that are being suppressed and how we're rescuing those neurons. If you now look in other organ systems, this is one of the key advantages that Inhibikase feels it has. So it's not as well understood by the lay public that Parkinson's disease actually has as its earliest manifestations certain forms of GI disturbance, difficulty in swallowing or dysphagia, difficulty with defecation, and so patients have very chronic constipation. And so when we go and ask questions about how do we introduce Parkinson's disease into a mouse and then ask the consequences of the introduction of that, we can introduce the plaque either, again, through an inherited or idiopathic model by introducing the plaque protein near the end of the stomach. When you do that, you actually see the migration of the plaque protein up the gut-brain axis known as the vagus nerve. It gets into the brain and then progresses through a path of pathology that very closely matches the path of pathology of the brain seen in the human disease. And again, that occurs over 1/3 of the animal's lifespan. So why is that compelling? Well, it says that the gut may even be an organ of origin for Parkinson's disease. And although that is not a fully agreed upon concept, and more importantly, we can now use the gut in conjunction with the brain in the clinic to diagnostically evaluate therapeutic benefit, to look at target engagement in the GI tract as a representative of target engagement systemically in the body. And that gives us very unique opportunities for defining what disease modification really means in Parkinson's patients, based on the fact that the GI and the brain are intimately linked through the disease process itself. So we've done experiments of this kind. You can give a red dye to a mouse, and you can evaluate the functionality of the entire GI tract from mouth to anus, measuring something called the whole gut transit time, which is essentially measuring how long does it take the mouse to process a food source and defecate it. If we give them a liquid dye, they form red pellets and defecate red pellets, and we can measure this whole gut transit time. So normal mice take about 165 minutes to process food and defecate. A diseased mouse at 3 months has a 300% effect. It goes to 500 minutes. A diseased mouse at 6 months has a 500% effect. It goes to 800 minutes. So it's very, very slow rates of defecation. So if we just look at 3-month data, again, looking just at the inherited disease model, the mice who are given the carmine dye take about 500 minutes at 3 months. But if we begin dosing at the end of month 2 and read it out just 4 weeks later, mice treated for just 4 weeks go from 500 minutes to an average of 219. So that's a more than 50% recovery. But in reality, what you see is that 90 of 96 measures are actually centered around 170. In our view, these mice are essentially completely returned to normalcy. And while we're not yet prepared to explain why this occurs, because we believe we do understand the process, but it has not yet been made public, there is one really important feature that I want to point out about what we understand about mechanism. So here you can now examine the GI tissue in the myenteric plexus itself. You can stain all the synuclein plaques that have been modified by ABL with an antibody against phosphorylated tyrosine 39 synuclein. You can see that there's a very, very robust expression of the phosphorylated synuclein throughout the tissue. We can stain the neural ganglia, but we were not able to stain the neurites in this image because of the age of the tissue, which didn't stain very well in the neurites. But we can look at the neural ganglia just to look at the associations, and you can see all the synuclein plaques that are ABL modified are intimately associated with the cell bodies, with the ganglia, and they're floating out here because they're all present in the individual neurites and neural axon fibers. So interestingly enough, if you look again at just 4 weeks of treatment at 2 different doses, in the treated animals, you don't see any of those synuclein plaque material whatsoever. So what we believe this represents is the first demonstration of seeing a functional recovery in an important organ of Parkinson's disease that occurs simultaneously with the clearance of the toxicity itself. And we think these results are really unprecedented. So what I've tried to represent today is that IKT-148009 drives functional recovery inside and outside of the brain. 148009 can drive clearance of what we think is the toxic form of alpha-synuclein, the ABL modified form. And more importantly, we think 148009, because of its GI and brain activity simultaneously in a living organism given once a day orally, will allow for use of the GI in combination with the brain to get unique paths to defining what clinical success means. So in our view, targeting c-ABL can block the path to neurodegeneration. And with that, I'd like to thank you for your attention and happy to take any questions.